Welcome to the Explainer. So today we're going to go inside the construction site of one of the body's most intricate systems, the GI tract. I want you to think of it like a massive building project. It starts with a really simple blueprint, but it ends up as this complex, fully functional marvel of engineering.
All right, let's kick things off with a clinical puzzle, one that you'll definitely see if you're in training. Have you ever wondered why congenital defects of the gut and the heart so often show up in the same patient? Well, the answer actually points to a shared and absolutely fundamental process way back in early development, figuring out the body's left and right sides.
Okay, section 1, the gut's first draft. We're laying out that initial blueprint. So to really get that connection between the heart and gut, we've got to go back to the very, very beginning. Every complex structure starts at the basic plan, right? For the gut, this whole thing kicks off in the 4th week of fetal life with just a simple, straight tube.
Now, this primitive tube, it doesn't stay simple for long. It really quickly differentiates into 3 distinct regions, and each one is destined to become a specific part of the final GI tract. You've got the foregut, which will form all the upper structures. Then there's the midgut, responsible for that massive length of intestines. And finally, the hindgut, which becomes, well, the final stretch.
And this whole process, it is not random at all. It follows a super organized pattern, moving in a predictable head-to-tail and then center outwards fashion. And the ones orchestrating this entire thing, well, those are the master control genes, things like the Hox genes. You can think of them as the construction foreman on the job site, literally telling each region exactly what it's supposed to build and become.
Section 2, twists and turns. This is all about the critical process of gut rotation. Because, let's be real, a long straight tube is just not going to fit inside a crowded abdomen. You can't just stuff it in there. So this brings us to one of the most dynamic phases of the whole project, how that tube folds, twists, and rotates into its final, very complex arrangement. Now, here's the crucial point. Think about our limbs. They're pretty symmetrical, right? The abdomen is the complete opposite. It is profoundly asymmetric, and that asymmetry is absolutely life-sustaining. Getting this right is everything. It depends on this incredible cascade of precisely timed genetic signals and even mechanical forces. And if this goes wrong, the clinical consequences can be just devastating. So how does this big twist even begin? It is truly an elegant dance between genetics and pure mechanics. First, a gene called PITS2 starts working overtime, but only on the left side of the mesentery. That's the tissue anchoring the gut. And this isn't just some abstract genetic signal, it causes real physical changes. The right side of that mesentery starts to expand, literally pushing the gut. At the same time, the left side condenses and gets stiffer, pulling it. This whole push-pull dynamic is what tilts the gut tube to the left, and that's what kicks off the entire rotation.
Look, when this process goes right, it's perfect. Everything fits just so, and the blood vessels have a nice, clear, secure path. But when it fails, you get what's called malrotation. Now the intestines are in the wrong place, and the vessels that feed them can twist on themselves, completely cutting off the blood supply. And I want to be clear, this is a true surgical emergency.
But here's where it gets even more interesting. This isn't just a story about genes. It turns out successful gut rotation also depends on metabolic pathways. Get this. Studies in amphibians have shown that if you just block the cell's energy factories, the electron transport chain, that alone is enough to cause malrotation. It's astonishing. This tells us that building a gut is also about metabolism and cellular energy, which kind of opens the door to all sorts of questions about how environmental factors might be involved.
All right, section 3, the duodenal hotspot. We're going to do a little case study in specialization. Let's zoom right in on one particularly tricky, specialized, and clinically super important region, the duodenum, which forms right at that junction between the foregut and the midgut.
So why? What makes the duodenum such a hotspot for things to go wrong? Well, it's because it is incredibly complex. It's not just a simple piece of pipe. It's more like a critical junction box, sort of the grand central station of the upper abdomen, where the stomach, the pancreas, and the biliary tree all have to connect perfectly. Any tiny miscommunication in the blueprint right here can have huge consequences. And clinically, we see this manifest as conditions like duodenal atresia, a blockage. It's a really common cause of intestinal obstruction in newborns. And its strong association with Down syndrome specifically is what has really driven researchers to hunt down the exact blueprint instructions that are needed to build a proper duodenum in the first place.
And this is where modern research is just incredible. Scientists can now create intestinal organoids,
We're talking mini guts in a dish, using cells from patients with rare genetic defects. And by doing this, they've identified a master regulator gene called RFX6. Seriously, think of RFX6 as the site manager for building the duodenum. And when that gene is mutated, the whole room gets built wrong. You get way too many mucus-producing goblet cells, not nearly enough of the hormone-producing enteroendocrine cells, and the whole thing just can't absorb fat properly.
So here's the clinical pearl, the big takeaway for anyone heading into a clinical field, especially GI. Knowing this blueprint, it is not just academic trivia. When you're doing a procedure like an ERCP, you are physically navigating this incredibly complex embryological junction, and understanding how it was built in the first place is what allows you to anticipate all the different anatomical variations that you are absolutely going to encounter.
Okay, on to section 4, wiring and plumbing. Time to install the vascular and neural networks. I mean, a building is useless without its wiring and plumbing, right? The exact same thing is true for the gut. It needs a huge vascular supply and a really intricate nervous system to do its job.
So what happens when that wiring installation goes wrong? You can get a condition called aganglionosis. Basically, these special cells, neural crest cells, are supposed to go on a long journey and migrate all the way into the gut wall to form the enteric nervous system. But sometimes they just don't make it all the way. And that leaves a segment of the gut, usually the colon, completely without the nerves it needs for motility. And that is the root cause of Hirschsprung disease.
And what's wild is that this single developmental failure, just faulty neural crest cell migration, is linked to this whole array of different clinical syndromes. We're talking about things that affect pigmentation, like in Waardenburg-Shaw syndrome, or even breathing control in Haddad syndrome. It's just amazing, and it really highlights how interconnected all of these developmental processes really are.
Now, let's talk about another key architect on this project, the hedgehog signaling pathway. Yeah, hedgehog. These proteins are responsible for building the villi. You know, those millions of tiny finger-like projections that line the gut and create its enormous absorptive surface area. Without hedgehog building those villi, you basically couldn't absorb nutrients. Okay, but here's where the story takes a really fascinating evolutionary turn. So you've got this ancient pathway that's been building guts for millions of years. What on earth does it have to do with breathing?
Well, it turns out that when vertebrates crawled out of the water and onto land, evolution needed a quick solution for separating the windpipe from the esophagus. So what did it do? It repurposed this ancient gut-building tool for a brand new job. It's a classic case of using an old tool for a new purpose. And there's a really cool theory that because this is a relatively recent evolutionary hack, the process just isn't as robust, and that might explain why defects like tracheoesophageal fistulas are actually more common than major defects in the villi, a process that evolution has been perfecting for a much, much longer time.
All right, last section, opening for business. It's all about the maturation of function. So the structure is built, the building is up, but now it's time to turn on the power. The anatomy's all there, but it has to become fully functional and ready for business right at birth. And functional maturity isn't like flipping a switch. It's a very gradual process. You know, immune cells start appearing pretty early on, neurotransmitters are there by 24 weeks, but that really coordinated sucking mechanism, that doesn't show up until 32 to 34 weeks. And this timeline right here is exactly why preterm infants face so many unique challenges with feeding, with motility, with host defense. Their gut just hasn't finished the job yet. And then, finally, at birth, the building is no longer empty. The first tenants arrive, the microbiome. This major phase of bacterial colonization begins, and let me tell you, this new microbiome is not just a passive tenant. It plays an active, absolutely critical role in training and directing how the immune system matures for the rest of a person's life. So this leaves us with a pretty provocative thought for the future. Remember those mini guts in a dish, the organoids? Well, researchers were actually able to use CRISPR gene editing to go in and correct that RFX6 mutation and reverse the defects right there in the lab. So my question for you is this. If we can now read the blueprint and literally fix the typos in a dish, how far are we really from being able to apply these blueprint edits to prevent or treat congenital GI disorders in actual people? It's a huge question, and it's right at the intersection of basic science and the future of medicine.